Hi, we're continuing with life science and cell biology in a nutshell. Um, Currently we're learning about a cell's part, or organelle called an endoplasmic reticulum. This, um, but this endoplasmic reticulum mostly, there's two types, rough and smooth. So the rough has ribosomes on it, on its cisternate, or the folds, so called. And the rough is contiguous with the membrane of the nucleus. So whenever the mRNA molecules come out of the pores of the nuclear pores on the envelope of the nuclear envelope, they come onto the ribosomes and proteins are synthesized off the mRNA chains. So, um, so the once the proteins are created on the ribosome, some proteins are delivered into the lumen, or the space inside the while others get groups uh, or sugar groups added to the proteins, forming glycoproteins. Um, so some let's see so some are processed within the in different ways. So some have metal groups added to them. And on the rough you can have four polypeptide chains brought together to form hemoglobin, for example. Um, Proteins are produced for the destinations of the plasma membrane, or the Golgi apparatus, or secretory vesicles, which secrete the proteins onto the exterior of the cell. Plant vacuoles for storage, lysosomes, endosomes. So lysosomes are for breakdown of various particles. Um, so and some stay within the Endosomes are for linking up of cells, and for communication and signaling of the cell membrane between cells, via binding of cells and channels between the cells, the membranes of the cells. So protein folding unit in the lumen of the rough the proteins get folded to produce highly important biochemical architectures which provide locking key and other recognition and linking sites. Protein quality control section in the lumen of the an amazing process of quality control checking is carried out. Proteins are subjected to quality control check, and any proteins found to be incorrectly formed or incorrectly folded are rejected. The rejects are stored in the lumen or sent for recycling for eventual breakdown to amino acids, the subunits of proteins. Protein structure and folding failures in the lead to problems. Proteins that are wrongly folded as a result of receiving an altered genetic message, or the incorrect, mRNA sequence, etc. These are never exported from the lumen of rough Direct connection between perinuclear space and the lumen of the allows for the movement of molecules through both membranes. So the membrane, perinuclear, means the outer membrane, double membrane of the nucleus, between the space that is between the space connecting the membrane, between the space between the the double layer. So molecules are able to move from the nucleus via the membrane into the ER space. So there's a connection between the outer membrane of the nucleus and the ER. The process of protein synthesis starts when mRNA moves from the nucleus to the ribosome. On the surface of the rough endoplasmic reticulum, the ribosome builds the amino acid chain. The chain is pushed into the cisternal space of the RER. When the proteins are complete, they collect, and the ER pinches off a vesicle. So the RER takes the proteins in the vesicle. The vesicle, which is a small membrane bubble, moves to the cell membrane or to the Golgi apparatus. Some proteins are used in the cell, some are sent out into the intercellular space, which is the space between cells, on the outside of cells. This is showing the endoplasmic reticulum and the folds or cisternae, which is connected to the outer membrane of the nucleus, the double layer, the bilayer of the nucleus on the outside. And the nucleolus is inside the nucleus. It's a region where ribosomal RNA, which are subunits of ribosomes, are made. These are chromatin strands, which are condensed versions of the DNA molecules. So this is a beautiful 3D picture showing the ER and the nucleus. So this is the system where by the genes are turned into proteins, which is very important for determining the structure and the function of organisms, whereby different genes encode different kinds of proteins. And also evolution occurs by the changing or selection of various genes encoding various proteins. So different proteins are created from different gene sections, different sections of the DNA or sequences. So when the genes change, or the sequence of DNA change, so do the proteins, and so do the functions of the different different organisms, because different proteins are created. This is an electron microscope image showing parts of the rough ER in a giant, in a plant root cell from maize, which is corn, so RER, and the dark spots are showing the ribosomes. Smooth ER, SER, is associated with production and metabolism of fats and steroid hormones, which are a type of lipid or fat. It is smooth because it is not studded with ribosomes. It is associated with slippery fats or smooth fats. So smooth ER is tubular versus the rough ER, and forms an interconnecting network or subcompartment of the ER. We'll continue in the next part. Thank you.